So welcome everyone to another wonderful journal club. Thank you to Malati for hosting, and thank you again to all our subcommittee community members, communications members, communications, Keke, Chanel, and Anke. And yeah, for sending out all the emails. So I think we're just waiting for a few more people to join. I look forward to the presentations from our two students. So we have the two students, master students, one from Northwest University, Chandre, and Kgalaletso, who will speak after Chandre from University of Johannesburg. Their profiles have been sent out to everyone, so you should have an idea of where they're from, what they do. And the idea for Journal Club is to have every month to have at least one session where we have students present their projects to the community. So the idea here is that everyone can have an idea what everyone else is doing at different institutions, and also to get feedback. So if any of you are interested in what the students are doing here, please get in contact with them. Perhaps the two students can just also put their details, the contact details in the chat box. Just put your email there. So if anyone wants to email them with some questions later on after the whole journal club, if you have a question later that you didn't think of now, you can always contact them, or if this is a project that you're interested in or something similar, please do communicate with them. The whole idea of this is to get communications going. So there is Chandre's email. And Kgalaletso, do you have your email as well, please? Okay, I think that's her email there from Fadel. Fadel posted her email there. There we go. So anyone has questions, please ask at the end of the presentation or email them. We encourage it. And for all future journal clubs, at least one session per month, we're going to have students present to the community. So Chandre, are you ready?
I'm ready.
Awesome. Okay, take it away for us. Thanks so much.
Okay, so here we go. So good morning, everyone. So as you know, I'll be discussing my MSc research on characterizing the immunometabolic profile of HIV/TB co-infection in cohorts untreated and treated for HIV. So I was supervised by Dr. Aurelia Williams, while Veronica Lees and Prof. Lewitz served as co-supervisors. So let's jump in. When infection with a pathogen occurs, a delicate balance is required so that the immune response is rigorous enough to effectively eliminate the pathogen while minimizing damage to the host, which may be induced by the very processes meant to protect the host. A major regulator of this balancing act is cellular metabolism. The attempts of the host to maintain homeostasis are ultimately reflected in the levels of intermediates of metabolic reactions referred to as metabolites, measurable indicators of the metabolic state of a patient. When immunity and metabolism meet, we get immunometabolism, which refers to the metabolic reprogramming that accompanies immune cell activation, proliferation, and effector function. Or in simpler terms, the study of the interplay between immune responses and host pathogen metabolism. The immunometabolic changes are mediated largely through small, soluble proteins referred to as cytokines. Cytokines are messengers of the immune system, measurable indicators of the immune status of a patient. So although the term immunometabolism has mainly been used in the context of the metabolism of immune cells, the effects of these alterations extend to systemic metabolism as the organism's energy and biosynthetic stores are directed towards the immune response. This is most prominent with chronic infections such as HIV and TB, since the immune system is continuously active for long periods. The acquired immunodeficiency syndrome, or AIDS, is caused by the human immunodeficiency virus. HIV infection is not curable, although antiretroviral therapy, or ART, can reduce viral load. On the other hand, tuberculosis is caused by Mycobacterium tuberculosis and is curable. So why do we care about these diseases? Tuberculosis was the #1 cause of death due to a single pathogen since 2007, but is now expected that COVID-19 will outrank it as of 2020. TB still caused 1.3 million deaths among HIV-negative people, with an additional 214,000 among HIV-positive people, accounting for a significant global HIV-TB co-infection burden. This burden is highest in sub-Saharan Africa, with South Africa having the second highest co-infection mortality rate in 2019, only outranked by Lesotho. As I have mentioned, TB is curable, but this is complicated in HIV-positive people as TB diagnostic tests are less sensitive, treatment is complicated, and the disease burden is increased, mainly due to the synergistic interaction between HIV and TB. Not only do HIV-positive people have a higher risk for developing TB, HIV and MTB amplify each other's effects. For example, the immune response to MTB increases the HIV replication at sites of infection, and HIV preferentially depletes MTB-reactive T cells. This results in increased systemic inflammation, immune deterioration, and dysfunction, increased disease burden, ultimately causing an accelerated clinical course. Thus, there is a need to better understand and characterize the complex immunometabolic changes induced by co-infection, since many deaths can be prevented with timely diagnosis and treatment. So although unstimulated, so that is without additional antigenic stimulation upon sample collection, blood cytokine profiles exist for HIV, TB, and the co-infected state, the literature is variable, and the co-infected cytokine profile has not yet been measured concomitantly with metabolic changes. In terms of metabolic profiles, large amounts of research exist for HIV and TB, with only a few limited and highly specific co-infection metabolomic studies. Multiplex cytokine measurements and untargeted metabolomics could aid in better understanding the immunometabolic profile and thereby the mechanisms underlying the co-infected disease state. This could lead to improved techniques for diagnosis, prognosis, disease management, and treatment monitoring, ultimately to improve outcomes for patients and better control of the pandemics. Therefore, the aim of this investigation was to use flow cytometry and two-dimensional gas chromatography time-of-flight mass spectrometry, or GCxGC-TOF-MS, to better characterize the HIV-TB co-infected state, as well as the impact of HIV and its treatment on a TB-positive population. So here you can see the aims and the related objectives laid out as part of the experimental design. We will take a closer look at the groups compared in a moment. I'm just going to give a brief overview of the methods we employed first. So firstly, the repeatability of both the cytokine and metabolomic analytical methods, as well as that of the apparatuses used, were ensured, and these validated methodologies were applied to all collected serum samples. For the cytokine analyses, serum samples were subjected to the Th1, Th2, Th17 cytometric bead array, or CBA kit. These Th designations refer to T helper cell types and the cytokines they typically produce. This is in essence a multiplexed ELISA, in this case to measure 7 cytokines concomitantly. To prepare the data for statistical analysis, missing values were replaced by 1/5 of the minimum value in the dataset for each variable. The dataset was then log transformed to achieve a normalized distribution, after which statistical methods could be applied. This included one-way analysis of variance, principal component analysis, and partial least square discriminant analysis. Similarly, for the metabolomics analysis, serum samples were prepared using the NWU's in-house total metabolome extraction and derivatization method. This method employs chloroform, methanol, and water as extraction solvents and methoxymation and silylation for derivatization. The samples were then injected in a randomized order on the GCxGC-TOF-MS for analysis. The resulting data was subjected to pre-processing, which included deconvolution, peak identification, and alignment using Chromatoph software. The dataset was then exported from Chromatoph and was normalized using total useful signal to reduce technical variation and make samples more comparable. The normalized dataset was subjected to various cleanup steps, including a 50% zero filter, batch effect correction, and QC coefficient of variation filter. The same statistical tests were applied as mentioned for the cytokine analyses, with the addition of a quantitative metabolite set enrichment analysis to identify biologically meaningful patterns. To identify trends in the datasets, Pearson correlations were also done within each dataset, but could not be performed between datasets since many of the cytokine levels fell below the limit of detection of the kit and hence were extrapolated values. So there was a bit of variation there, so we decided not to do the correlations between the cytokines and the metabolites. So for the fourth and last objective, the identified markers were interpreted biologically using previously published literature. Now we can take a closer look at the cohort. 4 groups were compared, from which 6 comparisons are possible. Those of interest to us are the following. Comparison 2, representing aim 1, the effect of untreated HIV-TB co-infection, and then comparison 4, the effect of untreated HIV infection on patients with TB, as well as comparison 5, the effect of HIV treatment on HIV and HIV-TB co-infected patients, and then comparisons 4 and 5 corresponded to aim 2. So samples that are untreated for HIV and or TB are difficult to obtain, hence the small sample size. However, studies in the absence of treatment are necessary as treatment induces another set of immunometabolic changes. Of the HIV-positive individuals, the majority already had AIDS, as seen by the CD4 T cell counts close to 200. Some of the patients still had relatively high counts, which accounts for the high standard deviation seen there. The cohort could not be described based on viral load, as this information was not available for all patients. So because of these limitations, the study is explorative and does not attempt to identify predictive markers, but it does contribute to better understanding the immunometabolic responses of the HIV-TB co-infected host and could direct future studies. As an entry into the results, let's first take a look at the PLSDA plots, which were used in an explorative sense here. So there's significant overlap between the disease groups in both plots, but note the patterns. In the cytokine PLSDA, the untreated co-infection samples, the purple dots, lie farthest away from the healthy samples, the grey dots, while the treated co-infected samples, the pink dots, lie anywhere in between the untreated co-infection and healthy samples in terms of their cytokine profile. In the Metabolomics PLSDA, on the other hand, the treated co-infected samples, the pink ones, lie farthest away from the healthy, the grey, with the untreated co-infection samples, the purple, lying anywhere in between in terms of the metabolic profile. So this is in line with what we know about the effect of ART on the immunometabolic systems of patients. ART typically results in an improved immune status. Hence, the treated patients are more similar to healthy in terms of cytokine profile, while ART typically causes metabolic aberrations in addition to those induced by infection. Hence, the treated patients lie farther away from the healthy individuals in terms of the metabolic profile. Based on the ANOVA results of the cytokine analyses, interleukin-6 and interferon gamma were significantly different between the healthy and in diseased groups, but there were no significant differences between the diseased groups. Thus, these results will mostly be discussed based on trends. IL-6 and interferon gamma correlated positively with each other, while IL-6 additionally correlated with IL-10 and interferon gamma with IL-17A. While CD4 T cell count decreased as IL-6 and IL-17 increased. Thus, IL-6 and interferon gamma, as well as IL-10 and IL-17 to a lesser extent, are potentially important in the HIV-TB co-infection context, as has also been indicated by previous studies. When the metabolomics data was subjected to ANOVA, 47 metabolites were identified that best describe the variation between the groups compared and fit into various categories. This is in line with previous blood-based metabolomic studies done on untreated HIV and TB patients, which has recently been reviewed by our group. So what did these results mean for the first aim, represented by comparison 2, the effect of untreated co-infection? This was elucidated by comparing healthy controls to the untreated co-infected group. From the metabolic ANOVA results, this metabolic chart was constructed. As we move through the most important aspects of this chart, the roles of the cytokines of significance will be explained where pertinent. Increased phenylalanine is a typical feature of HIV and TB metabolic profiles and is typically ascribed to decreased activity of phenylalanine hydroxylase, or PAH, which converts phenylalanine to tyrosine. Tetrahydrobiopterin, or BH4, is produced by the interferon-inducible enzyme guanosine triphosphate cyclohydrolase, or GCH, and is an essential cofactor for PAH. However, BH4 is highly sensitive to oxidation, and some immune cells produce neopterin at the expense of BH4. In light of the high levels of oxidative stress, immune activation, and the typically elevated levels of neopterin in HIV-TB co-infected patients, BH4 levels, and therefore PAH activity, may be low in these patients, which could explain the increased levels of phenylalanine. A large proportion of microbial metabolites were significantly altered in the HIV-TB co-infected patients relative to healthy controls. HIV infection causes impaired gut mucosal integrity, subsequent microbial translocation, and microbiome dysbiosis, which is also a feature of TB. And this is generally reflected in pathomorphological changes in the small intestine of HIV-TB co-infected patients. As many of these metabolites are also antioxidants, their decrease may also relate to prolonged oxidative stress. Furthermore, as IL-6 is predictive of anorexia or a decreased appetite in co-infected patients and correlates with immune activation as well as the extent of microbiome dysbiosis, the increased levels of IL-6 further support these microbial changes. HIV-TB co-infected patients have significantly increased tryptophan catabolism compared to patients with either disease state alone. Although we did not observe the typical pattern of decreased tryptophan and increased kynureninins, we did observe decreased tryptophan with decreased levels of gut microbe-derived tryptophan metabolites, which can also be produced endogenously in an IL-4-dependent manner. The decrease in catabolites can be explained when recognizing the antioxidant nature of these metabolites, the high levels of oxidative stress, and decreased IL-4 levels observed here. This indicated that these metabolites may be important in the co-infected state, in addition to the kynurenine-tryptophan ratio, which has previously been suggested as a co-infection biomarker. Mannitol was another microbial metabolite of interest. This metabolite cannot cross membranes, and as such, its increase in blood has been associated with increased gut permeability. Some other metabolites have been associated with the presence of NTB, of which the increase suggests an increased bacterial load in co-infected patients. There were also metabolites that could not be linked to known biological processes and were presumed to be of exogenous origin, that is, originating from the environment. Returning to the metabolic chart, notice the number of altered amino acids, lipids, and energy metabolites indicated in purple. These changes indicated decreased energy availability in the co-infected patients. This fits in with the previously mentioned poor state of the gastrointestinal tract of these patients, and previous reports of a higher risk of malnutrition and more severe cachexia, which is the unintended loss of muscle, and in some cases, fat mass associated with disease in co-infected patients. Specifically, the increased levels of 3,4 and 2,4-dihydroxybutyric acid, metabolites that have been associated with the feeling of satiety, were interesting in the context of cachexia. The mechanisms of cachexia are not well understood during HIV and TB. So to investigate how these metabolites may relate to that, 3,4-dihydroxybutyric acid and lysine, one of the most significantly altered amino acids, were correlated to all other significantly altered metabolites. Although these metabolites generally correlated with metabolites relating to increased oxidative stress and inflammation as well as to each other, their relationship to cachexia and body composition changes require further investigation. Moving on to the second aim, which involved firstly the effect of HIV on TB, elucidated by comparing untreated TB patients to untreated co-infected patients, and secondly the effect of ART by comparing untreated co-infected patients to co-infected patients receiving ART. As many of the concepts of importance here have already been mentioned, I will only highlight aspects of the effect of HIV and its treatment not yet mentioned. The trends observed in the cytokines when comparing TB patients to co-infected patients could be linked to known effects of HIV on TB. These trends suggest a failure of immunoregulation, as regulatory and pro-inflammatory cytokines are concomitantly elevated, without showing significant improvement once ART is started. This supports previous reports that although the levels of T cells increase upon ART, the heterogeneity and functionality of these cells are not fully restored. Glucuronic acid is involved in xenobiotic detoxification and was reduced in the untreated and treated HIV-TB co-infected patients relative to the TB group. Various members of the gut microbiota can cleave glucuronic acid from its conjugated xenobiotic or steroid during detoxification, although this is variable among species. Thus, if the composition or functionality of the microbiome was altered in such a way as to decrease the number of species able to perform this cleavage, the levels of unconjugated glucuronic acid may be altered. Furthermore, increased and chronic xenobiotic detoxification due to the leaky gut caused by HIV, as well as the continual detoxification of ART in treated patients, contribute to the reduced levels of this metabolite. Cadaverin was detected in reduced amounts in the untreated co-infected patients relative to the TB patients, and even further reduced in the co-infected patients on ART, although not significantly. Since the gut microbiota produces cadaverin, the reduction observed here is most likely ascribed to the altered gut health and microbiota composition in these groups. Cadaverin forms part of the glutathione metabolic pathway, and as such, its reduction may also be associated with oxidative stress, which is known to persist even during ART. So in summary, HIV worsened the immune activation, inflammation, energy crisis, and gut mucosal damage and microbiota dysbiosis, as well as oxidative stress experienced by TB patients. While ART improved some effects of HIV, the gut mucosal damage and oxidative stress showed minimal change or were worsened. In conclusion, IL-6 and interferon gamma was shown to be important in the HIV-TB co-infection context, and the observed trends further suggested a failure of immunoregulation. The roles of IL-10 and IL-17 during co-infection warrant further investigation in light of these trends and those reported in previous literature. The metabolic profile of the HIV-TB co-infected individuals shows significant overlap with profiles previously described for HIV infection and TB, suggesting considerable energy distress, which is reflected in those metabolites associated with increased amino acid and lipid catabolism and partially explained by high levels of satiety-inducing metabolites. These metabolites also suggested a mechanism by which cachexia could be mediated in HIV infection, TB, and HIV-TB co-infection. The large proportion of the altered microbial metabolites suggests that gut damage may be more severe during HIV-TB co-infection. This may be mediated by and contribute to increased immune activation, inflammation, subsequent oxidative stress due to concurrent HIV and MTB infection. As some of these metabolites could be related to MTB directly, these results also reflect the increased success of MTB during co-infection. Overall, these results suggest that HIV partially augments the HIV-MTB synergy through its detrimental effects on gut health, affecting energy availability. Thank you for listening.
Thank you so much, Chandre. That was a very impressive presentation and a very thorough master's project, very in-depth. So does anyone have questions for Chandre? You can type it in the chat box here, or you can raise your hand and ask verbally. Chandre, I have one question for you. Do you have any plans to test your hypothesis that you have created through your master study? Do you have any future experiments that you're going to try?
So thank you, Dr. Shane, firstly, but not me, myself. I'm working on another project now for my PhD, but there are a few other master students in the pipeline who will be answering some of these questions. So
okay.
Yeah, there's a lot to study.
There's a question in the chat box. What was your rationale behind using different N values for your experiments?
So I think the N values were the same for most, for both the cytokines and the metabolic experiments, but we could not, like the study, I wasn't involved in the design of the study, so I can't really answer how the samples were collected and determined how many of each. I think the samples were provided by a clinic, so we used what we had. So that's why some of the groups, there were not the same amount of people in each of the groups. And like I said, untreated samples, especially co-infected samples, are difficult to obtain because people are put on treatment the moment they test positive. So I don't know if that answers the question.
I think it does, because I see you have a lot of involvement from Desmond Tutu Foundation and UCT, IDM, all over. So it's a very large project. And you used the samples that were available, I believe.
Yes, yes. It is unfortunate that we couldn't have larger groups because that would have made the entire study more useful. But yeah, it is difficult to get these samples, and so when you have them, we just use the opportunity to get some preliminary information.
Of course, I'm sure everyone will be interested to see how COVID-19 will fit into your trifecta infection analysis.
Yes, that would be very interesting to see.
Well, this is already complex, just having two diseases together. So having a third one into it is going to be even more complex. Sure.
Yes.
So I don't think there are any more other questions. So if anyone has any other further questions later on you might think of, please do contact Chandre. I'm sure she'll appreciate any interest in her project and she'll love to chat about it. Thank you again, Chandre, for such a wonderful presentation.
Thank you, Dr. Shen, for the opportunity. Thanks to everyone for listening.
Okay, so we're going to then move to our next speaker from University of Johannesburg. Is she ready?
Yes, I am. I'll be joining from Dr. Vidal's laptop.
Okay, no problem. So you can present your slides.
Yeah. Can you
There you
guys
go.
see?
I can see them, yes. So when you're ready, go ahead.
Okay. Good day, ladies and gentlemen. My name is Kgalaletso Othibeng, and I'm a master's candidate at the University of Johannesburg under the supervision of Dr. Vidal Tigizimana. So the work that I do in my master's focuses on the exploration and use of computational tools in metabolomics, as well as investigating the effects of biostimulants on plants under normal and stress conditions. Sorry. Okay, so what exactly are plant biostimulants? So plant biostimulants by definition refer to substances which when applied to plants stimulate various natural processes for enhanced nutrient uptake, crop quality, and plant tolerance to environmental stresses. So these biostimulants have been categorized as being either microbial based or non-microbial based. As the name suggests, microbial based biostimulants contain microorganisms such as fungi and or bacteria. And examples of these include PGPR as well as trichoderma. Non-microbial biostimulants, on the other hand, do not contain microorganisms, but are rather bioactive substances which can be formed from microbial metabolism, as well as through biological and chemical transformation of plant and animal matter. Some of the examples are shown here. So humic substances, phosphides, and silicon, to name a few. Although the use of biostimulants in agricultural practices has positioned itself as a more eco-friendly and sustainable strategy for improved crop health, stress resilience, and ultimately improved crop production, majority of the studies that have been done on these biostimulants have mainly focused on the observed physiological phenotypic effects, and very little is known about the cellular and molecular events that underlie these physiological effects. That is, there is a lack of knowledge in regards to the mode of action of biostimulants. Omics sciences, such as metabolomics, offer unique opportunities to interrogate the plant metabolism under specific physiological conditions. Thus, in this project, we applied metabolomics to investigate the effects of biostimulants on plants in order to provide the knowledge that is needed in the biostimulants industry. For example, the generated knowledge here can be used or can inform the formulation of biostimulant products, as well as the implementation in agronomic practices for sustainable food development or food security, sorry. Metabolomics can be defined as the data-driven study that involves the qualitative and quantitative study of all the metabolites that are present in a biological system, such as a cell, a tissue, or an organism as a whole. And as explained in the previous journal club, the workflow of a metabolomics study involves several steps. And I had a pointer quickly, sorry. Involves several steps, such as there's the study design, planting, and treatment step. This is very important, and the research question on the study should be considered here. Secondly, there's the sample preparation step, which involves harvesting or sample collection, as well as metabolite extraction. Next, the extracts are analyzed on analytical platforms, such as mass spectrometry or NMR, and the step is also referred to as the data acquisition step. The acquired data are then processed and prepared, followed by data analysis, and lastly, biological interpretation using pathway and network analysis. So, despite the advancements in technologies used in metabolomics platforms, there are several bottlenecks at different steps of the metabolomic workflow, which limit the biological insight that is generated. And one bottleneck of particular interest in my project is the metabolite annotation. So majority of the signals that are detected in metabolomic studies are known, and this is referred to as the dark matter. And this can be attributed to the complex nature of metabolites, as well as the limited coverage of libraries and databases. So to try and address this challenge and improve on metabolite annotation, several computational tools are currently being developed, and these include the 4IR-inspired tools and workflows such as classical and feature-based molecular networking, MS2LDA, and MolNet Enhancer. And these aid in data visualization, mining of spectral data, and fragmentomes exploration, as well as allow for improved metabolite annotations. So to explain how molecular networking works, I will use this workflow on the left. But first, I need to mention that the principle of molecular networking built on the assumption that metabolites that are structurally related or are similar will produce similar fragmentation patterns when fragmented. So this means that spectral similarity is used as proxy for structural similarity. So looking at the workflow, you have a sample of pooled metabolites which undergo MS-MS fragmentation, giving rise to the respective MS2 spectra. Then using the MS cluster algorithm, identical MS2 spectra will be collapsed into single nodes as shown here. This is then followed by pairwise spectral alignment, whereby the query MS2 spectra are compared against each other in order to form molecular clusters where metabolites with similar spectra will be clustered together into what is called molecular families. On the right, I've provided literatures to read which explain and also explore the use of these molecular networking tools. So as mentioned in the program statement, there's a lack of understanding with regards to the mode of action of biostimulants. Therefore, to illustrate how these tools, be it molecular networking alongside pathway analysis, can help understand the mechanism of action that is employed by biostimulants, I will use one of the papers published in our lab where we looked at the metabolic choreography of maize plants treated with a humic substance-based biostimulant under normal and starved conditions. So briefly, the study was designed to comprise 4 groups, namely control 1, which were the starved plants with no HS treatment, control 2, which were the non-starved plants with no HS application, and the HS treated plants under starved and non-starved conditions. So for metabolomics analysis, the metabolites were extracted from the maize leaves using methanol extraction, and the extracts were then analyzed on LC-MS analytical systems. Moreover, different methodologies were applied to mine and interpret the generated metabolomics data. These include computational tools such as molecular networking, which is used for annotation, as well as metabolic pathway network analysis. So in our study, we applied molecular networking to get an overview of the extracted metabolome. This figure over here represents a feature-based molecular network, which was computed using the feature quantification table and MGF files obtained from the MS-DAL processing software. The nodes in the network represent MS-MS spectra of pooled metabolites in the extracted metabolome, and these can be connected to one another by edges as shown here, forming clusters of structurally similar metabolites called molecular families. So as mentioned in the introduction, molecular networking builds upon the assumption that structurally related metabolites produce similar fragmentation patterns when fragmented. The same principle is also followed for metabolite annotation, during which each of the query spectrum will be searched against reference spectra in the GMPS house libraries in order to assign a putative annotation to the metabolites that have library matches. That is, for a query spectrum to be matched to a reference spectrum in GMPS spectral libraries, the MS-MS spectral similarities between the 2 spectra must be above a user-specified threshold for structural similarity, which in our study was set to 0.7. So from a total of 3,792 nodes in the feature-based network, only 87 heads were found. So that means that there were only 87 query spectra which were putatively matched to GNPS library spectra, and these are represented by the colored nodes with bold black edges. Examples of these include DGMG, which is a glycerolipid, as well as chlorogenoquinone and caffeoylquinic acid, which are ACA compounds. So one of the advantages of molecular networking is that if one node in the network is annotated, the unknown nodes in the same cluster can be manually annotated. Please keep note that the assumption is that the nodes that are in the same cluster are structurally similar. So, for example, with the ACA cluster over here, shown in C, caffeoylquinic acid was putatively annotated using molecular networking. And from this, we were then able to manually annotate two other ACA compounds in the same cluster, namely caffeoyl glucarate with an MZ of 371 and caffeoyl hydroxycitric acid with an MZ of 369. These three compounds are similar in that the core structure is caffeic acid and differ with respect to the organic acid or the sugar that is conjugated to caffeic acid. Furthermore, to explore the chemical space or profile of the extracted metabolome, MolNet Enhancer was applied, and this workflow combines the output from molecular networking tools such as feature-based, MS-tool-DA, which is a substructure annotation tool, and in silico annotation tools such as NAP and dereplicator plus. And this is done in order to enhance the chemical overflow through chemical class annotations. So from our obtained results, the metabolome that was covered included unknown classes, which means they had no matches, and these are shown in gray, as well as putatively annotated classes such as ACA compounds, glycerolipids, cinnamic acid, and derivatives, to name a few. Moreover, as illustrated by the chord plot in D, a total of 75 metabolites were annotated, and these belong to various classes. This was done both manually as well as using molecular networking. So to then situate the changes in the annotated metabolites in a biological context, metabolic pathway analysis was performed using an overrepresentative analysis or a method of the metaboanalysis bioinformatics suite. So pathway analysis makes use of preliminary knowledge for the analysis of metabolite patterns from an integrative point of view. That is, it maps the matched or annotated metabolites rather into known KEGG pathways. So for my results, the key pathways that were revealed by pathway analysis included the linoleic metabolism, the serine biosynthesis pathway, the cysteine and methionine metabolism pathway, and carbon fixation. So without discussing everything, I'll just take one pathway that is linked to growth promotion. So the alpha-linoleic pathway shown in B is involved in the biosynthesis of oxylipins and the signaling hormone jasmonic acid. So as shown by the pie charts in the pathway, maize plants that were treated with the humic substance biostimulants under normal conditions showed an increase in levels of oxylipins. These are indicated by the orange side of the plot. So examples are OPC and OPDA, which is oxophytodianoic acid. And although the mechanistic roles of these individual oxylipins are still poorly understood, some of the general functions of oxylipins in plants include modifications of chloroplast function, plant senescence, stomatal conductance, and antifungal and antibacterial activity. Furthermore, the oxylipin pathway also leads to the generation of phytohormone jasmonic acid. So other signaling metabolites such as indole acetic acid, shown here, and salicylic acid also showed an increase under conditions of or under the presence of HS treatment as compared to the control. These phytohormones are involved in various biochemical and physiological processes in plants such as seed germination, seedling growth, stomatal aperture, and respiration, as well as in interactions with the environment. Therefore, the measured changes in the lipids and hormonal networks in maize plants suggest that HS biostimulants remodel the maize metabolism towards growth promotion via the activation and enhancement of physiological events for improved plant development. So under nutrient starvation conditions, heat map and pathway analysis were also done to investigate the effects of HS treatment. So looking at the heat map in A, the relative concentration analysis revealed that amino acid and phenolic compounds, so that's the ACA derivatives and flavonoid profiles of the stuffed plants shown here, are similar of the stuffed plants treated with HS. Sorry, this is the middle one. They were found to be similar to the profiles of the non-stuffed plants shown here. Amino acids were significantly reduced due to nutrient starvation in non-treated plants, thus suggesting that there was an increased degradation of these amino acids as an alternative mechanism to compensate for the limited nitrogen and or carbon supply. Enhanced amino acid degradation is usually observed in plants that are suffering from carbon deficiency. However, with the application of HS, there was an increase in these metabolites as compared to the stuffed conditions without HS. And this could mean that the HS biostimulant either directly supplies the plant with carbon and nitrogen, or it triggers other mechanisms which will provide the plant with sufficient nitrogen and carbon. Several studies have also shown that treatment with HS enhances nutrient acquisition and mobilization. So looking at the linoleic acid pathway shown in B, the application of HS also showed a differential response in the oxylipins under stress conditions. So under the stress conditions, the oxylipins were found to be, so under stuffed conditions, some increased, but then also decreased under conditions with HS treatment. So the functions of oxylipins have been found to be stress signal transduction. These are also found to be involved in the regulation of stress-related gene expression and interaction with other hormonal signaling pathways. So overall, this metabolic alteration suggests that HS application under nutrient stress conditions induces metabolic readjustments to alleviate the negative effects of starvation in plants. So to compensate the pathway analysis results, MetaMap network analysis was employed. This web-based tool is able to map all the detected metabolites into network graphs using the KEG reaction pairs KRP database and the Tanimoto chemical similarity between PubChem substructure fingerprints, and thus generating an overview of the metabolic regulation under specified conditions. So as infographically depicted on these networks, there are three main metabolite clusters, namely the phenolic indicated in circles, the lipids shown by the arrows, and the amino acids shown by squares. These metabolites were mainly interconnected based on chemical similarity shown by the green edges, and the KEG reactant pairs interactions in green are observed or seen between amino acids, as well as the two oxylipins OPC and OPDA. So the overall metabolite networking shows the overall changes due to HS starvation in the metabolite space. So A shows the stuffed plants without HS, while B shows stuffed plants that were treated with HS. To compare the two, the amino acid cluster shows an overall decrease in levels in the plants that are starved without HS treatment, as compared to the overall increase that are observed in plants that were starved, but also treated with HS. This could point to possible regulatory events that underlie HS-induced metabolic reconfigurations in maize plants towards growth promotion and alleviation of nutrient starvation. To summarize, metabolic reconfigurations related to HS biostimulants induced growth promotion involved different alterations in the levels of amino acids, phenolics, hormones, and lipids. And these were translated into physiological events such as membrane remodeling, improved chlorophyll content and photosynthesis rates, improved nitrogen and carbon assimilation, and elongation of roots. The main HS mechanisms that were involved under stress conditions included metabolic or cellular homeostasis, low-cost machineries in response to starvation, and increased nutrient uptake and assimilation. So in conclusion, the study showcased the use of computational tools and their benefits thereof in mining spectral metabolomics data and improving metabolite annotation. Also, the knowledge that was generated from this work provides a morphophysiological and metabolomic gateway to the mechanisms underlying the effects of HS biostimulants on plants. Such insights lay a foundation for the advancement of the biostimulant industry and the incorporation of these formulations in agronomic practices for sustainable food security. These are my references. I'd like to acknowledge the following people: Dr. Fidel, Ms. Lerato Napali, as well as the MSA for the opportunity, and the biochemistry department. Thank you. Are there any questions?
Thank you so much. Wow, that's a very good presentation. I think I'll have to go back to my presentations and improve a little bit. So you guys, both of you have set the bar very high with very good presentations, very complex studies. And I think us as South Africans, we are way too modest on what we're doing. So as master's students, you guys are doing complex studies and complex science, and are publishing on it. It's excellent. And I think it's very cool the way you guys can annotate unknowns through your molecular network. There's a question in the chat box from Vuisani. So this study is quite technical, great talk. Do you think there might be a synergy between the endogenous biostimulants and the usage of fertilizers for plant growth? Please excuse my ignorance.
Thank you for your question. So if I understand your question, it was like, is there what's the similarity between the use of fertilizers and the use of biostimulants? Right? So fertilizers also provide nutrients that are needed by plants, but then the main point of adding these biostimulants is to reduce the use of these fertilizers, because most of them are chemical fertilizers and have been shown to have detrimental effects on the environment. So the biostimulants are usually used together with the fertilizers that can be applied together. So the biostimulants do not replace the use of chemical fertilizers. I guess I can say it complements it.
Okay, I hope that answers the question. Anisha has her hand up. Go ahead, Anisha, you unmute.
Hi, thanks. I'm struggling to type my question out because I don't understand quite how these biochemical stimulants are applied to the plants, but as with fertilizers and pesticides and those kind of things, do you know if there's any carryover from these stimulants into the final product, you know, into the human metabolism? And is there any research into the effects that that might have on human metabolomics?
No, there have not been, or I do not know of any studies showing any human, any effects of these on human metabolism. Sorry.
I think that's down the line in the future. So there's some comments here, excellent presentations, amazing. So that's wonderful. Well, very well presented, an excellent study, wonderful presentations from both students. Thank you and good luck. So indeed, thank you so much, both of you. you have made a great beginning for our student presentation, a great bar that's been set for everyone to try and emulate the same results, the same presentation. So please, if anyone has any other questions, you can ask now. We have another 10 minutes left of the session. So raise your hands or ask the question. Or you can have the emails presented earlier on. If both are presenters, can you just write your email one more time at the end here on the chat? So if anyone has any questions lingering later on, please contact our presenters. We want you to communicate with them, to discuss their projects, get involved with them, give feedback, maybe some collaborations going on. I see Malati is sharing a screen here. So on the Metabolomics YouTube site, might be what Malati is trying to sell here, is all the past presentations. I think the recording will be there. So go to YouTube and look for Metabolomics South Africa. Is that correct, Malati?
Yes. We'll try and upload these as soon as the sessions end. So in case there's a part you want to run over or make notes.
Thank you for that, Malati, record keeper. There's a question from Ilsa. Are these plant metabolomics methods provided as a commercial service? Oh, I think we lost our speaker. Fidel or our speaker, Kgalaletso?
Sorry, what was the question again?
Are these plant metabolomics methods provided as a commercial service?
Not yet really like formally, but yes, if someone has samples or is studying to seek and design, can contact relevant people in metabolomics, plant metabolomics, and then set plan or ways to do those studies. For analysis of the samples, also the same apply. Contact the relevant people, and then where fees are required to carry the experiment can be done. Yeah. So
also follows up this. She says that we may get many requests from CHM, the Center for Human Metabolomics. We get a lot of requests for plant metabolomics. So once it becomes commercialized, I think we're going to refer a lot of people to you.
Okay, that's fine.
Vuyisani has another question. Maybe I missed it. How do you starve these cells, and for how long do you starve them?
Okay, so the staff plans were provided with only 40% of the nutrient solution, and this was done after emergence. And the treatment of H's was then applied at 4-leaf stage. Harvesting was then done 3 days after. So starvation was done by providing the plants with 40% nutrient solution as compared to the non-starved, which were given 100% of the nutrient solution.
Okay. So I want to share one thing before we finish up. For our next session next week, I have this voting system going on. It is anonymous, so you can vote without fear of your name being there. So it's just a topic of human health for next week from the Nature Journal series. So these are all the most recent metabolomics papers from the series on human health. And some of the suggestions here are given on the screen. And as you can see, we are a tie across 5 votes. So I'm going to put this in the chat box. So everyone, I do encourage you to come and vote for a paper next week. So we're going to do a new emerging paper on human health. It ranges from microbiome and metabolome of students sitting in front of the screen. So those are people in front of the phone, iPad, TVs, PCs. What is the effect on microbiome metabolome? So this would be interesting. A more metabolism focused one of excess adenosyl methionine inhibits methylation by catabolism to adenine. One on aging and muscle function. So that's also important in human health. Gut microbiota, crosstalk. And I'm sure a lot of women will be interested in this. After you diet, why do you gain weight back? And so I know my wife, she has a lot of answers to this. Yeah, we have a vote. And the last one, exercise, how it affects non-alcoholic fatty liver disease using non-targeted metabolomics. So please go vote here. I'll put this link on the chat here. So it's anonymous voting, and we will select a presenter for that for next week. Human health, new emerging research in metabolomics in the Nature Journal series. So I'll ask the communications group to send that possibly at the end of tomorrow, regarding which topic wins, because at the moment we just have one vote extra on the last one. So please vote on your preferred topic. And the recording for this will be on YouTube. So just go to YouTube and search for Metabolomics South Africa, and you'll find the recording there. Thank you again to both our presenters. They did a wonderful job for us. And next month we'll have more students presenting. And thank you for everyone for joining and the questions that were asked. Great questions there. Wonderful involvement with the questions. I love it. So let's hope we can continue this going in the future journal series. So thank you to everyone. And I hope to see you next week.
Goodbye.
Bye.